Yesterday we talked about transcription and translation and the genetic code, right? So the DNA triplet code coding for amino acids, which then make up the polypeptide, and how that is a redundant or degenerate code and it is non-overlapping, and then how the DNA is first transcribed into messenger RNA that goes into the cytoplasm, uh, attaches to ribosomes where tRNAs bring in amino acids corresponding to the triplet code on the mRNA, and right, peptide bonds are formed between amino acids to form the polypeptide chain. And then there is mass production of polypeptides because you have multiple ribosomes attaching to a single strand of mRNA to produce multiple copies of that polypeptide. And again, that depends on what the utility of the polypeptide is. It could be a neurotransmitter, so you want more of those available, right? For example, or digestive enzymes, and so you want obviously more than just a single molecule of the enzyme. Today, we'll first talk about gene mutation, and then we'll sort of shift over to talking about plants. So 2C1 gene mutation, right? And so we know DNA has information, it has instructions for how the cell functions and what the cell is, right? So whether it is a liver cell or a cell in your eye, right? Or the skin, and right, what functions it has to carry out, what the proteins that are being made over there, and everything else, right? And so all of that is dictated, so to speak, by the code, the nucleotide base sequence on the DNA. What can happen, right, is a change can occur in the base sequence, and that is called a mutation, right? So a change in the DNA, right? Now it could be of different types. It could be a point mutation, right, or a simple gene mutation. So we've talked about point mutations, and we'll talk a bit more about the different kinds of point mutations. So it could be a point mutation where, right, the nucleotide sequence is affected, so the nucleotide sequence is changed. It could be a chromosomal mutation, which involves changes in the positions of genes within the chromosomes. And so, and obviously, if the gene is not where it's supposed to be, then that is going to have an effect on its expression. And then you've got whole chromosome mutation. And this is where entire chromosomes are either lost or duplicated. And so we know chromosomes are basically, right, the whole strand of the DNA coiled up. So entire chromosomes are lost or duplicated, right? And this normally happens, well, only happens during meiosis, right? Because that is when DNA replication occurs. Well, it does happen during mitosis, but right. Uh, so I correct myself, right? It can happen during mitosis, but not so much, right? Generally, more so in meiosis. And right? The problem with if a mutation occurs during meiosis, so meiosis is only happening in the gametes, right? And so these are the reproductive cells. You could say that it is the most damaging, right? Why? Because that mutation will be passed on to the offspring. So something going wrong in your gametes might not affect you, right? But if fertilization occurs and the zygote forms and a new individual begins developing and there's a mutation in either of the gametes, then it could be very damaging, right? Because it's passed on, and that's obviously something that you know we wouldn't want. So an example of that happening, right, of whole chromosome mutations happening in the gametes, right, is Down syndrome. Down's syndrome, where the individual, the offspring, ends up with 3 copies of chromosome number 21. And so normally you have two, you have a pair, right? And so this is an example of whole chromosome mutation. And so they have 3 copies and they show very visible characteristics about their facial features and their mental growth and their speech and everything, right? So right, in general, mutations are either, right, generally mutations are either neutral, right, which is mostly the case in point mutations, right? So it depends on the type of mutation, or detrimental, right? So even in point mutations, it can be detrimental. Chromosomal mutations and whole chromosomal mutations, definitely, right? So there aren't cases that I know of, at least, where you've got whole chromosomal mutations and they are neutral. Okay, there are certain cases in point mutations, and we actually discussed them uh, in our last class, where there was no change, right? And that's owing to the fact that the DNA code is redundant or degenerate, right? Okay. And it is very rare. It is very rare that a mutation would, give properties that have an advantage, that give an advantage to the organism in which the mutation has occurred, right? So it's very rare. We could say we might have a few examples, but again, the rarity, right, is it's like very occasional, very infrequent. Okay. Now let's talk about point mutations a bit. And so mutations can occur in 2 ways. First, let's talk about that. Mutations do occur naturally. So mutations do occur naturally when DNA is replicated. And so, it could affect a nucleotide base. So that could be changed or in some way, or the DNA might break, for example. And so there are natural repair mechanisms, as I've already mentioned in the earlier class, right? We already talked about that. And so their repair occurs also naturally, right? But there are certain cases where it's not able to be repaired, and so uh, right? That would lead to problems happening. So an example of a nucleotide base sequence being changed, the frequency of that happening is very low, right? So it's 2.5 into 10 to the power 8 per base. So it's very, very low. So that would, actually be like 0.000000025, right, per base. So it happens, right, once in, right, like 10, 2.5 times in like 10 million times. So every 10 million nucleotide bases, maybe there's a chance of one nucleotide base going wrong, right? Then mutations can also occur due to external factors such as X-rays. Oops, not those X-rays. X-rays, right? So it's not like you're going to get an X-ray at the hospital and you're going to have a mutation and you're going to turn into a superhero like Wolverine or Cyclops or someone, right? But repeated exposure, and that is why the technician operating the X-ray machine wears a jacket that has lead in it, because lead does not allow X-rays to penetrate through it, and so that's protection for the technician, because he or she is operating the machine on a daily basis, conducting numerous X-rays in a single day, and so the level of exposure that is experienced by the technician is far higher, obviously, than what you would experience maybe having one X-ray per year, even if it's a per year, although right, you're not having an X-ray every year. So, right, so you've got X-rays, and then you've got ionizing radiation, and an example of that is UV, right? So repeated exposure to UV can cause mutations, and that is one reason why, right, in some countries skin cancer is a major problem, and there could be certain chemicals that cause mutations. And so all of these are given the term mutagens, right? So if you're familiar with the old school cartoon, Teenage Mutant Ninja Turtles, they were regular turtles and they turned into these humanoid turtles, anthropomorphic, right, creatures who learned ninja and became superheroes because of mutagen. That's not how it happens, but they were mutated, and so right, they probably came into contact with some chemical that changed them. Right, so exposure to mutagens obviously increases the rate at which mutation occurs, because these promote mutations. Okay, now coming again to different types of point mutations that can occur. Right, and so the first type that can occur is a substitution, which we've talked about yesterday. And so, for example, you have the sequence TAG, sorry, TAA, TGC, and that changes to TAG, TGC, right? So this is what's being changed. Now, there is a likelihood that this is not going to have any effect because of the degenerate code. So it might code for the same amino acid, but it also might not. So we're not saying substitution mutations, sorry, are safe, right? As we'll see an example of sickle cell disease, which you're probably familiar from your IGCSE as well, that it is a point mutation that causes problems. right? In fact, before talking about the rest, let's talk about sickle cell disease. And so what's happening over here is the code, right? It happens in hemoglobin. So sickle cell disease, right? And the code for hemoglobin, right, is mutated. And so what happens is that you've got the codon, the triplet code GAG, which codes for glutamic acid, and that is changed to GTG, right? So the second nucleotide base in the triplet, and this codes for valine, right, or just VAL. And the problem for that is, if you recall, right, there are hydrophobic and hydrophilic interactions, and so this guy is hydrophilic, whereas this guy is hydrophobic. And so if you end up having a hydrophobic amino acid where a hydrophilic one should have been, and it's going to cause havoc as far as the protein folding is concerned. And that's exactly what happened, right? So the protein, the hemoglobin folding is affected. And it cannot carry oxygen efficiently. Right? The shape of the red blood cell changes. And they tend to clump together. So there's a, danger of blood clots and all of that. And it is life-threatening, right? The condition is life-threatening. Any questions?
No, sir.
Okay, next up is a deletion point mutation. And so as the name suggests, right, you have a nucleotide base that is removed, right? And so you again, right, so that same sequence, you have TAA, TGC, and now you've got TATGC, and so you're missing this, right? So you're missing the third base, for example, right? That one's gone. And so the problem that, right, happens is that initially you had the codon TAA and TGC, and so they're coding for corresponding amino acids, or the stop codon perhaps. But now what you have is TAT and then GC and whatever's after that. So, right, there's a shift, right, in the triplet base. And that's not going to affect one amino acid, that's going to affect the entire protein. And right, it's definitely going to produce faulty, right, either non-functional or detrimental, right? So maybe they're producing something that's causing harm, right? Even being non-functional is going to have an effect. So if you've got a non-functional example enzyme, right, and so whatever reaction it's catalyzing is going to be affected, and that's going to cause problems. But even if it could make something that's causing harm in other way, right? So in either case, a deletion is not good at all, right? It's because the degenerate code, right, cannot make up for it. And the third is an addition or an insertion, and this has a similar effect to the deletion, right, because it shifts the entire code. And so you again have the TAA TGC, and you're fitting in another nucleotide, and so you're getting TAA TAGC, right? So you have this extra guy over here, right? And so you've added that, and again, so the entire code has been shifted. So you've got TAA, which is fine, but now you've got TAG, and then C, and then whatever, right? So again, you've got a shift. And it's going to cause the similar faults. All right.
Yes.
So what we learn definitely is that gene mutations affect phenotype. DNA mutations, right? Whether the point mutations, chromosomal mutations, or whole chromosomal mutations, right? They affect phenotype. Phenotype, just to recall, right, are the physical characteristics, are the right that show up uh, based on the genotype, which is the genes and the sequence and the proteins and everything, right? So an example obviously is the sickle cell disease that we just talked about, right? Because it is physically changing the structure of the hemoglobin, the structure of the red blood cell, the shape of the red blood cell, and its oxygen carrying capacity. Then right, there are other diseases, for example, thalassemia. So let me get that spelling right. Thalas double AEMI, right? And so this is a genetic disease that affects the blood proteins. And so blood proteins are not manufactured properly. And then you got another cystic fibrosis. And so this affects the function of membrane proteins, certain membrane proteins. Now, right, with all of that, right, most mutations don't cause anything, right? So again, they're neutral, so to speak, right? And the reason for that is, right, mutations, we talked about 98% of the DNA is non-coding, right? It's not genes. So unless it's like a regulator for a gene, right? If it's a non-coding region, so mutations in non-coding regions of the DNA will not have any effect, right? And I don't know, can I sort of take that? Do I have anything else to add to this page? Not really. So I can fit it in over here somewhere, right? Oops. So mutations in non-coding regions of DNA will not have any effects. And we are done with talking about gene mutations, right? So there's slight details and elaborations in the chapter, so you can take a look at that. Any questions?
No, sir.
So how about we move on to talking about plants?
Sure.
So we are jumping forward to section 4A, unit 4, section 4A, and we have chapter 4A1, which talks about the cell wall. Right, and so they give the introduction that plants are also eukaryotes similar to animals, and so right, they share many of the features and organelles that you would find in an animal cell, such as the different membranes and the cytoplasm, nucleus, rough and smooth endoplasmic reticulum, Golgi apparatus, the mitochondria, right? But there are certain fundamental differences, right? And one of them are the cellulose cell wall. Okay. And so they give the analogy that a plant cell is like a balloon filled with jelly inside a cardboard box, if that helps your understanding anyway. Right, and what they're trying to tell you is that the balloon itself is the cell membrane, and the jelly is the protoplasm, so the cytoplasm and everything else. And the cardboard box, which is obviously stiff compared to the balloon and the jelly, is the cell wall. Right, and so what's basically protecting it's giving structural support, it's protecting the balloon, the jelly filled balloon is the cardboard box, and so that's the analogy they're giving. Okay, so the cell wall, right, provides strength and support. Right, and right, for plant cells, so plant cell walls are made up mostly of cellulose. Okay, so the plant cells are made up of mostly of cellulose. It is a carbohydrate, a polysaccharide. In fact, it is a polysaccharide of beta glucose. So if you just recall when we talked about biological molecules, beta glucose is an isomer of glucose, right, where the position of the OH group is different on the first carbon, right, as compared to the alpha glucose, right? So it's the position of the OH groups. And so it's a polysaccharide of beta glucose, It forms straight chains and it is insoluble, right, which is obviously helpful because the medium inside living organisms is mostly water, it's aqueous, and so you don't want the cell wall to dissolve, right? So it needs to be insoluble, right? It is a rigid uh, molecule, and so that obviously helps with the function of providing strength and support. What else do we have? Right? It is freely permeable. So it plays no role in controlling the substances passing into out of a cell. Right? That's not its job. So it is freely permeable to water and to pretty much everything else. Okay. In certain more mature cells, right, substances such as suberin or lignin, depending on which cells they are. Let's correct that. Lignin are added to the cell wall, right, which changes and basically reduces their permeability. And right, in the cells and in the regions and the tissues of the plant where this happens, you want it to happen, right? So, for example, lignin is added to the xylem vessels. We're familiar with xylem vessels, right? And so those are the vessels, the tissue that is responsible for the transport of water and everything dissolved in it, right? So all the ions and the minerals and salts and everything. And so the walls of the xylem vessels are lignified, right? And so that helps because if it were freely permeable, there's a high concentration of water in the xylem vessel. And so before it reaches the leaves, for example, right, it's going to leak out on the way. And you don't want that happening. Okay. Now, right, let's talk about how the cell wall is built up. So the structure of the cell wall. And so, right, the first layer is called the middle lamella. So when it's built up initially, and obviously it's built up after cell division, so mitosis. Right? It is mostly made up of pectin. The name is the middle lamella. It is made up of pectin mostly, right? And pectin is also a polysaccharide. And right, on a side note, when fruit juices are made, pectinase is an enzyme that is used to treat the pulp, which digests the pectin into its units, so disaccharides or monosaccharides, and that you know helps utilize more of the fruit pulp in making juice and adds to the sweetness, because we know polysaccharides are not sweet, mono and disaccharides are sweet. Right, so on a side, and so this pectin acts like a glue, right, holding cell walls of adjacent plant cells together. And the thing about pectin, it has a lot of negatively charged carboxyl groups. Right, and these attract positive calcium ions. And so that forms a substance called calcium pectinate. Right, or well, actually calcium pectate, not pectinate. Pectate. Right, so this binds the cellulose. So right, the cell wall doesn't start with cellulose, it starts with pectin. It binds the cellulose. Now moving on, right, cellulose microfibrils and matrix build up on both sides of the middle lamella. Of the middle lamella. Let me add middle. And so initially, right, the walls are very flexible. And all the cellulose microfibrils are arranged in a similar direction, right? And this is called the primary cell wall. All of this is the primary cell wall. Now, what happens next is a secondary thickening, as the plant ages. And so a secondary cell wall builds up. With the cellulose microfibrils. So over here, all the cellulose microfibrils, right, were in the same direction. And that gave flexibility. Over here, the microfibrils are placed at various angles. Right, so they're going everywhere. And so you can sort of imagine that that's going to reduce the flexibility. It's going to make it, much more rigid. And that is what we want. Right? And so they're built up with, a dense layer. Then there's a substance called hemicellulose which is added to it, which makes it even harder. And then we already mentioned about, substances like suberin or lignin being added, which increases the thickening and the strength and the hardening, right? So that is, how the plant cell wall is built up. They go over how the cellulose molecule is built up using, beta glucose monomers. I'm just going to show it to you before I, move on or perhaps wrap up. Let's see what happens. So yeah, and so as you can see, right, there's a difference between alpha and beta. So in the alpha glucose molecule, the OH group on carbon number 1 is below the plane, so it's down, whereas in the beta glucose it's up. And so, right, what's happening in cellulose is you're having a polymer of beta glucose, but what happens is that every other beta glucose molecule is sort of inverted, right? So you got this one that's the right way up as it should be, and the second one is flipped, right, 180 degrees around the x-axis, right? It's called inversion. So it's flipped 180 degrees around the x-axis, and so you can see, right, this carbon number 6 over here, that's like a peg sticking out. It's down here. The oxygen is down here. So it's the same molecule, just inverted it. And so the OH group that was down here on carbon number 4 is now up and aligning with the OH group of carbon number 1. We're going to have a condensation reaction and a glycosidic bond forming, right? And so the next beta glucose molecule will be just fine as it should be, right? So this one's inverted, this one's regular, this one's inverted, this one's regular, and so that's how the chain will grow longer. And then between the different cellulose molecules, there's hydrogen bonding between the OH groups, which are slightly negatively charged, and the oxygen, right? And so yeah, so right, the OH groups, the hydrogen actually becomes slightly positive because of the polarity of the oxygen, right? And so that is going to form a hydrogen bond with the oxygen that is part of the ring of the glycosidic bond, right? And yeah, so right, that's how these hydrogen bonds keep the cellulose molecules sticking together. They are 1,4 glycosidic bonds, right? They're 1,4 glycosidic bonds as given over here. And most animals, including humans, do not have the enzyme available to break these bonds, right? Even cows and other ruminant animals do not have the enzymes themselves. It is the bacteria in their gut that is able to break those bonds, and so they are able to utilize plant material and cellulose as food, right? For us, it acts as roughage to improve digestion and peristalsis, but we cannot digest plant material, right? We cannot digest cellulose. So something we already knew, and so these different cellulose molecules come together to form microfibrils, and so that's an electron micrograph of cellulose microfibrils. The last thing I want to talk about, right, for this chapter, I'm not going to touch plasmodesmata, is something we already know, right? So in a cell wall, there are 10,000 or 100,000 cellular molecules which form microfibrils, and they're deposited in a matrix of hemicellulose. So we've talked about all of that. There are other short chain carbohydrates in the mix, such as mannose, xylose, and arabinose, and right, they mix together to make it gel well and more rigid, and keep it tight and all of that. And so all of that together makes what's called a composite material, right? And as you already talked about earlier when we talked about osmosis, right, in plant cells, the cell wall helps with the turgidity of the cell, which keeps the which keeps the cell firm and helps with the vertical position, the plant standing upright. And if water leaves the cell, it becomes flaccid, and so the plant is drooping, it becomes floppy, as they have this the word they've used is floppy. And so, right, the structural support is very important for the plant's overall structure, right? It's not just the structural support of the individual cell, but the structural support for the entire plant as a whole. Any questions?
No, sir.
Are you sure?
Yes.
Okay, so I will stop there. We'll continue with this topic the next time, inshallah. And yeah, that's all for today.
Thank you, sir. Allah Hafiz.
Allah.